have you guys found the animal that is hidden within here? It is definitely a dog. Does anybody know what kind of dog that is? Bonus points if you do. It is a Great Pyrenees. Good job. I have one. I have a Great Pyrenees and a Norwegian Outcast. And that's what that puppy is too. Okay. So, infectious disease. Does anybody know what kind of organisms fall into this category? Or what infectious disease is? You guys are missing one there. Olivia, you're missing one. You got 3 out of 4 of them. So corona is a virus, so it does fall into there. Protozoa. What's another word we can give to this? Protist. Very good. Excellent. So bacteria, viruses, fungi, and protists that are parasites would be illnesses caused that fall into this category of infectious disease. So I worked as a research scientist and lab manager at the University of Pittsburgh Graduate School of Public Health Department of Infectious Disease and Microbiology. And that was right out of college. We had a really good, awesome opportunity. I was friends with a lead manager of the lead lab, and they had a startup lab. So it was a PhD who was starting his lab, and he was looking for a lab manager. And for this interview, what I had done to prepare is I had read all of his research, and I actually had made a connection with research that he was researching a particular protein in bacteria. And I had read other research about trypanosomes, which is what my research was on. And I made the connection that maybe since he was researching this particular protein in bacteria, and it was a well-researched protein in trypanosomes, that maybe that's why he wanted to make the transition into trypanosomes. And I had just made that connection myself by reading his publications. So I had asked him during my interview, is just out of curiosity, is that why you're doing this research? And he says, that's exactly why. And he was like, do you want the job? So do your research before you go into an interview. I was hired, since I was the first person hired, he hired me as the lab manager. I didn't really have the credentials that a lot of other lab managers had to be able to hold that position because I was right out of college. A lot of times you would need to get experience to be able to move right into a lab manager position. So I was fortunate, and I worked there for 3 years, and it was a really incredible experience. And I'm going to tell you all about it. So what I researched was the Trypanosoma brucei. It is in the domain Eukaryota. Can you identify what kind of organism it might be? I guess that was me in the picture. Right there. Out of one of my degrees. I had multiple, but it was out of my last one. So could you take a guess at what kind of organism this might be? Oh, Mason got it. Yes, a protist. So what kind of protist? I know you can see the text, but you can't see there. That's what I'm getting at. Parasitic. Thank you, Mason. That's what I was getting at. It is a parasite. So what does a parasite do? Is it helpful? What does it do? Right. So it's not a beneficial organism. It is an organism that is going to cause harm to its host. Um, and it falls into the category of protist. So it is an eukaryote. It is a protist. And something really unique about the trypanosome that I will show you guys soon that it doesn't have with any other organism is that it is in the class Kinetoplastia. And that's because it has a very special organelle that no other organism alive has called the kinetoplast. And I'm going to show you a little bit about what that is. So the specific species that I researched was Trypanosoma brucei. And I did want to say on the other side that I forgot to say some benefits and cons to working in a university. So benefits are great. You get to uh, go and pursue any other degrees virtually for free if you wanted to continue your education. Usually there's no cost associated with that, or the costs are minimal if you are an employee of a university. And there's lots of other perks, especially working at the University of Pittsburgh. I was able to get into like the Carnegie museums for free. That was very cool. Also, your children will get a humongous discount on their education if they choose to attend that college. And also, we followed the holiday breaks that the students followed. So if had like a really long Christmas break, we also had a long Christmas break. That was really nice. Some cons, though, to working on university level is that when you are researching at the university level, you're typically researching off of grants, and grants are limiting. If you're getting an echo, it's probably because of me because I'm logged onto two devices. I will be logged off of that soon. I apologize. So the cons are that whenever you're working under a grant, it has limitations because what you're being paid is based off of the grants that you have. So sometimes it can get kind of tight with salaries because you are only able to expend so much to salaries. Um, nope, I had the screen like that on purpose. So I wanted to show you guys the morphology of the trypanosome. So does anybody know what morphology means? Morphology. What is the morphology of an organism? Right, so when we're looking at the morphology of an organism, we're looking at its structures, so its structural components. Okay, so I want to draw out the trypanosome. Once I figure out how to do it, hit this button. Let's see if that does it. Oh, it's very light. It only gives me gray, red, yellow, green, and blue. We will go with the pink. All right. Come on. All right. So this is a general outline of the way that a trypanosome looks. So a trypanosome has that very special organelle that I told you guys about, and that is called the kinetoplast. And that is located right here at the tip by its head. And the kinetoplast is a very special organelle. It contains mitochondria, circular mitochondria DNA, and it only has this single mitochondrial DNA organelle. So where we might see multiple different mitochondria in other organisms, the trypanosome only has a single mitochondrial DNA organelle. So that's right at the top, and then attached to it, we see a flagella. Does anybody know what a flagella does? What is the purpose that a flagella serves? Thank you, Saoirse. Yes, it helps you to move around. Yes, so this is the flagella. And then the flagella is actually connected to the body. It has like this flap in it. So these lines are kind of to represent the connection between the flagella and the body of the trypanosome. And we call that an undulating membrane. And then in the middle, it has a nucleus. What would you find inside of the nucleus? Excellent. And then you will find also a nucleolus in there. And then, of course, inside of here, this is where we see the cytoplasm. So those are the main components of the trypanosome. It also has another flagella in the back of it. Okay, so I wanted to show you guys the structural morphology of the trypanosome so you can get an understanding of what that kinetoplast does. And now that we know what the taxonomy and morphology is of the trypanosome, I would like to watch a video about its ability to parasitize. This is a very cool video. However, I'm worried about, I think I'll move this one. I heard
A brutal killer invades a traveler's blood.
He said it was very, very critical that you get to medicine that quickly, otherwise you're dead. I've been to Africa 11 times. We were there 3.5 months ago, and I was there for a little over 3 weeks.
But 2 weeks into the trip, Dwight suddenly starts to feel unwell.
I just started to feel very, very weak. One day you wake up and you're so weak that you can't hardly get out of bed. I just got weaker and weaker and weaker until I was so weak I couldn't stand on my own.
What's astounding is that the illness that has Dwight fighting for his life started with a simple fly bite.
Africa is a big place, and there's different animals, different pests in different places. But in Tanzania, you have the tsetse flies. They are about the size of a housefly and normally a little bit bigger, but they're just extremely aggressive, and you just hate to have them pestering you all day long.
But the tsetse fly that bit Dwight wasn't just a nuisance. It was infected with a deadly parasite, a single-celled killer called trypanosoma. And when the fly bit him, These parasites flooded into Dwight's bloodstream. Inside his body, these cunning intruders began to divide and elongate. They used their long tails, called flagellum, to swim throughout his bloodstream. The result is a severe case of trypanosomiasis, or African sleeping sickness. Dwight's immune system is helpless to stop the microscopic assassins. Each individual trypanosoma parasite is armed with a shield of proteins. In the bloodstream, white blood cells recognize these proteins as foreign, and they build up antibodies that attack the proteins. But trypanosomes can actually change their protein coat of armor, rendering the blood's antibodies useless. This leaves them free to reproduce and devastate the body's red blood cells, the very cells that carry nutrients and oxygen throughout the body. Starved of nutrients, the patient goes into a coma and ultimately dies. A brutal killer.
That's one of my favorite shows ever. It's called Monsters Inside of Me. I love that show. But that's a really cool short clip of what happens to a person that becomes infected with the trypanosome. So this is how you spell it. It's trypanosomiasis, African trypanosomiasis. It is amazing and terrifying. I agree. I love it. It's one of my absolute favorite shows. So this disease is caught in sub-Saharan Africa. So sub-Saharan Africa. Saharan, we learned about bottoms. These are grassy lands, hot grassy lands. And the sub in there means that it's going to be in the lower portion. So sub-Saharan Africa is actually a pretty widely distributed location on the African continent. And the tsetse fly is the vector. So a vector is an organism that carries the parasite that then infects another organism. So the tsetse fly, it actually lives around rivers. It likes to live around really, really hot rivers, and it just swarms there. And a lot of people in Africa do actually rely on those rivers for life. They rely on them for food, for water. So a lot of people will, the cheese part, I missed that. So a lot of people will go to the river and they'll be swarmed with these tsetse flies. And something that's really unfortunate is that in Africa, the hospitals are not really distributed as close together as they are in a lot of other countries. This is considered a third world country. So the hospitals you can see are more widely distributed. So if you're living in the main part of Africa, you're going to see a lot of hospitals close together. But if you're living over here, or over here, over here, what happens is that these people sometimes have to walk because a lot of these people don't have cars, okay? So they'll have to walk sometimes for days to get to a clinic and to get treatment. And the effects of the trypanosomiasis show within about 1 to 3 weeks after you first have the bite from the tsetse fly. So by the point that they actually get to the clinic, sometimes it's too late based on where they live. And that's very unfortunate. And it continues to get more unfortunate. So the way that they test to see if you have African trypanosomiasis is by a simple blood test. It's actually just a rapid test. They take your blood and it can actually determine if you have the trypanosomiasis really quickly, which is awesome that they have this test. But like I said, you know, sometimes they have to walk for a really, really long time, and they don't see the symptoms sometimes until 1 to 3 weeks after. And by the time that they actually get to the clinic, they will have been heavily infected with the parasite. And the most unfortunate part of this story is the treatment. So, this is what they used to treat people who have African trypanosomiasis: arsenic. Yes. There's actually an arsenic form of a drug. It's metoprolol or something. That's a complicated word, but it's arsenic. And that's because of the trypanosome. So I'm going to do one more drawing and then I can turn this thing off. So if you remember, whenever we saw that video of the trypanosome, they showed that it has what's called a glycoprotein coat of armor, okay? So it has this coat of armor that wraps around it, and it serves as armor. And it's very, very hard to penetrate this armor. And even the body's antibodies and the body's own immune system, whenever it comes in to attack, it's useless, because this trypanosome, it has another really unique function, is that it can just keep on creating these coats, just multiple coats, just over and over and over. Every time that it is hit, it just creates a new coat. And the only thing that they have found for a very, very long time to be able to actually penetrate this armor was arsenic. And I can turn this off now. Let me clear this. There we go. And arsenic poisons a lot of people that get the drug. So there's a lot of people who were, no, there's no vaccine. So a lot of people who go to these clinics to get the arsenic treatment die from arsenic poisoning. A lot of people. So up until about 10 years ago, they had no other drug. That's primarily what they used. And there was another drug that was introduced about 10 years ago, and it's just not as effective to treat the stage 2 of the African trypanosomiasis. But it does help really well in stage 1, whenever there's just not as many of them. But whenever the body is really overwhelmed and there's a lot of trypanosomes, still arsenic is the #1 treatment. And people die from arsenic poisoning. It's a very, very serious disease there. So, what were my daily tasks like whenever I was a scientist? That's kind of what I would like to show you now a little bit, and then I'd like to talk a little bit about what my particular research was. We have 15 minutes left, so I should be able to get through that. So whenever I came in the morning, my lab was considered a level 2 biohazard lab. It goes up to level 3. Level 1 is where you'll find bacteria, not as serious, a contagion. Level 2 was where I was at, and level 2 was because the trypanosome can be researched in 2 different forms. It's researched in its procyclic form, which is considered its insect form. So when it's in the gut of the tsetse fly, it's considered in its procyclic insect form. And then there's the blood form, whenever it's actually inside of the person. And they are 2 different forms, and they have 2 different biohazard levels. So whenever I was working on the procyclic form, which a lot of our research could be done on the procyclic form, it didn't need to be done on the blood form. And when I was working in the procyclic form, there were certain precautions that I would have to take based on that level of infection that would be possible to me. But whenever I worked with the blood form, then it became a biohazard level 3 lab, and that was in a different room, and I had to take different precautions. So therefore, when I was working with that form, I would have to be in a full suit. Um, my friend who worked there, she worked on HIV, and every single time that she came in contact with it, she would have to be in the full suit. So you'd have to be fully covered, kind of like those hazmat suits that like the nurses wore during COVID. You have to be in the full like hazmat suit, completely covered, whenever you're working in the biohazard level 3. The very first thing that I did whenever I walked in every single day was I'd go to the incubator and I'd get my media and my cells, and I'd have to like do this to them. I have to shake them. And this kind of keeps them going and keeps them alive, because the incubator that we have wasn't a shaking one. There are some of them that like do shake for you, but the one that we had wasn't a shaking one. So I'd have to go in every single day like this. The disadvantage of that part of it is that since I was a lab manager, I had to come in every day and do that multiple times a day. So on Saturdays and Sundays, I had to drive to the lab and go like that to the cells. Yeah, it was a little annoying, that part of it, but it was part of my job though. So something else that I would do if I wanted to start a new experiment with fresh cells, I would go to the liquid nitrogen container. So this is where we deep freeze cells. And this is kind of what it looks like whenever you're pulling them out of the tank. And you have to put these very thick gloves on to pull them out of the liquid nitrogen. This is what deep freezes the cells whenever you're starting a new fresh media of cells. I'll show you in a minute here what media means. So this is my hood. This is typically where I do all of my research, okay? So the hood is for safety, okay? The hood is going to include my media. The media is what allows the cells to grow. It's going to include my pipettes, all of my tubes, my flasks, anything that I need to work with, any of my drugs that I'm using. They're all going to be, I'm going to bring them, I'm going to store them in the refrigerator or the freezer at a certain temperature that is suitable to life for that organism. And then I will, or the incubator, and then I will bring it to the hood whenever I actually need to work with it. And then this is kind of how you work under a hood. You would be in a full lab suit, and then you would have the gloves on. So this is whenever I would be working with the procyclic form, so the biohazard level 2. I would have to take those extra precautions in the biohazard 3 whenever I have to be full suited up to work with the blood form, because the if the procyclic form were to somehow to have touched my skin, it wouldn't infect me. So therefore, you just have to take extra precautions with the blood form because there's a higher risk of infection with it. So this is typically how you do work whenever you're working under the hood. So I spent a lot of time under a hood like this, working with pipettes like that. So kind of this is what it looks like a little bit whenever you're working with the pipettes. So you would get your frozen cells and you would add your media with the pipette, and then you would add it to the flask. And then if you have any drugs that you're adding, you usually will titrate and add the drugs to the tubes. And then you would take like a small amount of that drug and then add it to your flask. So this is kind of a daily thing for me that I would do in the lab. So really fast, just a couple of experiments that I did for my publication, Western blotting. I'm not going to get into the whole entire process, but let me just tell you, it was my favorite. I loved doing these. They were so much fun. I don't know the other science teachers in here if you guys have ever done a Western blot, but it's so much fun. You load in the electrophoresis, the DNA electrophoresis, and then it runs through a gel. And then you have to put it in this machine where you're breaking up the proteins, and then you transfer it to a kind of like a photo paper, okay? And then at the end, what you do is you take that photo paper from the gel. So the gel will actually touch the photo paper. And then the photo paper will actually go through a like a photo processor. So we'd have to like go in the dark room and like process the images. It was so much fun. I loved doing these. But I'll tell you what, though, if I knew that I had to run a Western blot, I knew that I had a 15-hour workday ahead of me. So this is a workday that I knew that I was going to be there really late. But it was a lot of fun. I really liked doing these. PCR is another experiment that I did a lot whenever we're amplifying the amount of DNA that we need for an experiment. You would run it through a PCR machine. Something that I did the most in terms of actual research for the publication would be working in the microscopy lab. So the microscopy lab is actually located, I worked at Pitt, and Pitt is multiple different buildings, okay? So I worked, my main lab was in the Graduate School of Public Health, but the microscopy lab was like several buildings over, and like, if you know University of Pittsburgh, it's a lot of hills. So I'd be walking up and down giant hills every day, like going to the different labs that I had to go to. So I would go to a lab that kind of looked like this. And there were very, very expensive microscopes there. And then we would work on a screen like this. So you would actually do your experiment right there in the microscope lab. And then so I would take all of my materials with me and I would do the actual experiment there. And then I would go into where the microscopes were. And then I would sit at a computer like this and I would capture as many images as I could. And the really cool part of this part of the job is that they were always living whenever I did this. So I would be watching them squirm around like crazy. It was super, super, super cool. And here is a picture that I actually took. This is an immunofluorescence. And what I was doing was I was actually staining. Can you guys guess what I was staining here? If you were to take a guess, what was I doing an immunofluorescence on? Here, let me zoom in a little bit here. What would you guess? No, it's called immunofluorescence. Immunofluorescence. So what immunofluorescence is it is a certain type of experiment where you're basically highlighting a certain type of tissue or component of a cell, okay? So you're actually injecting certain types of immunofluorescence and they are going to attach to different parts of the cell. And then they're gonna light up like this. So what I was looking for in this particular experiment, I was highlighting the cytoplasm, and then I was highlighting the kinetoplast and the nucleus, okay? So the cytoplasm is orange here, and the kinetoplast and the nucleus is green. And the purpose of doing an experiment like that was because I was trying to look at the cell cycle, because the cell cycle in the trypanosome, it goes through this different form where the kinetoplast will double. It replicates via binary fission. So the kinetoplast will double and the nucleus will double, and then it separates like that, okay? So if you were to look at my experiment compared to the cell cycle, you can actually see the cell cycle taking place. So you can see that there's two kinetoplasts here, and then there's one nucleus. You can see here there's two nucleus. So this is showing different stages of the cell cycle. So that's what I was looking for. So the immunofluorescence is just an experiment that allows you to visualize this. So that was the purpose of this experiment. And I would spend hours and hours and hours and hours and hours at this microscope lab, very late days whenever I knew I had to go to the microscope labs. So ultimately, what was my research? And if you notice on my paper, it does say Pamela Bookite. That is my maiden name. But the purpose of my research was to look for a drug target. So what is a drug target? A drug target is basically some part of the cell that a pharmaceutical drug can attach to and cause some kind of change, okay? So what we were looking at was a very specific protein that was involved in the cell cycle. Okay, so what we were trying to do, Katie, no, this is Katie Ekman. Katie Ekman actually was a friend of mine in college, and I did outreach, and I would go to my previous schools, and I would show them the research that we were doing, and Katie was a student at one of my previous schools, and she attended one of my presentations, and she said, "Can I please just come and observe you?" And she came one day and just didn't leave. We ended up hiring her as another lab tech. So that's how she ended up getting in. Yeah, but she ended up just coming and just staying and just basically like doing free work for us until we hired her. So what we ended up finding in our research is that the particular protein that we were looking at was a good candidate to be a drug target. So our research is very, very small in terms of the big picture of what we wanted to accomplish, right? So what we ultimately wanted to accomplish was a way to have a drug that wasn't going to kill so many people, because arsenic was killing a lot of people. And our research was just one small tiny piece of that puzzle. We just found a protein that we were able to prove would be a good drug target. And once the research is out there, then, the pharmacies then, decide if they want to work with that drug target or not, and see if they can test it out and come up with a drug that might be successful. But this is really how it starts. You do research like this, and then you publish it. So why am I teaching now, and why am I not working in infectious disease anymore? Trypanosome does not infect people in the United States. It is very hard to get funding for. It's very hard to secure grants to research this particular organism. And a big reason for that is because the trypanosome is a very unique class of organisms, because it has that kinetoplast. And it actually requires very specific media, and like everything that you have to use with it for cell culture is really, really, really expensive. And ultimately, we did not secure another grant, and the lab ended up having to close. But I got to be a part of something really cool. Okay, here's a picture of my little girl in the lab. It was a lot of fun. You can tell I miss it. This is Kylie though, playing with the pipettes. It was a lot of fun. And something I do with my class all the time is I have an animal of the day. So of course the Trypanosoma brucei is going to be my animal of the day, but I also wanted to give you guys a challenge animal of the day, and that's its cousin, the Trypanosoma cruzi, which causes Chagas disease. So if anybody wants to learn something cool today, look this up, because this one is a trypanosome, but it's found in America. ... Look it up yourself. You guys will be blown away by this one. Does anybody have any questions? That is all for me. Any questions from anybody? If not, then thank you very much for coming to my presentation. I hope that you learned something great. I hope that you have enjoyed biology this school year. It's been so great being able to teach you all these really cool and fun things. And for my students, I will see you guys back on the last day of school. We're going to do something special on the very last day. So I hope you guys all pop in that day. And I hope that you guys have a great day.